Hi, my name is Tusita. Welcome to my biology class. Today we're going to do Cambridge AS Paper 1 class, which is a MCQ paper. And here you can see the today's MCQ question. So what we're going to do, we're going to start from the first question. The first question is, when making measurement in experiment, which method could have a parallax error? So it's basically this one, the parallax error. So what is parallax error? Uh, okay, parallax error is when we're going to measure something. The exact measurement is shifted due to a different viewing angle. So what does it mean? So let's say if we have a different viewing angle from top, from bottom, and that's a correct way. So if this is the right measurement, so when we're going to measure, we might get this one, or if you're going to measure this one, you're going to get this one. So that is actually the shifting the correct measurements due to the viewing angle. So when we measure this one from this angle from the bottom, so this is the reading. So we're going to make this one, so this is the reading. So that's a parallax error. So this parallax error, it's a very common when we're going to measure in a cylinder, or maybe let's say it's a ruler here. It's a ruler. It's same again like this. So you have problem here, you have problem here. That's a ruler. That's a measuring cylinder. So let's go to the choices. Using a calibrated eyepiece graticule to measure length, using a measuring cylinder to measure volume, using a ruler to measure length of a shoe. So it's very clear here, we use the ruler here and we use the measuring cylinder here, but microscope, the calibration of eyepiece and graticule, which has no effect because we directly look always from this angle and there's no angles from microscope. So then there won't be any errors using microscope. So then measuring cylinder and 2 and 3. So C is the incorrect answer for this one. So we go to the next question. Which part of the cell is often continuous with the rough endoplasmic reticulum? Okay, rough endoplasmic reticulum, can draw this one. That's a nucleus. Nucleus has two membranes. First membrane envelope and the second membrane. So that's a nuclear envelope. And you should not forget that we have a small like this kind of called nuclear 4, and after that the outer membrane is going to continue like this, to make the rough endoplasmic reticulum, and then you can see the smooth endoplasmic reticulum. So this is an endoplasmic reticulum, and endoplasmic reticulum rough one is always associated with the ribosome sitting on it. That's a structure, and this is a no ribosome, a smooth endoplasmic reticulum, and that's a rough endoplasmic reticulum. So the choices are cell surface membrane. Now cell surface membrane is something here I can draw here. There's no continuation. Golgi apparatus, Golgi is like this. So Golgi has no direct connection, physical connections. Mitochondrion, okay, mitochondrion again I'm drawing like this. So it has no again connection. And next one is a nuclear envelope. So here with the mitochondrion no, and with the cell membrane no, with Golgi no. Yes, here there is a direct connection with the nuclear envelope. So that's the answer. D is the answer for this question. Here we go to the next question. The next question is question number 3. Which structure are found in both chloroplast and mitochondria? So just quickly we try to learn chloroplast and the mitochondria structure. So I can show you first, let's say chloroplast. So chloroplast, something like this structure, and slightly bigger than mitochondria maybe, and then we have this structure called thylakoid. So thylakoid is connected with the other thylakoid with middle lamellae, and it has the two membranes, inner and outer membrane. So that's a stroma, and that's a thylakoids, and that's a inner membrane. I use IM, outer membrane OM, and it's a stroma contain mostly the ribosome and maybe starch granules. That's a stored food as a ribosome, and like this circular DNA. So this ribosome is 70S. So usually the other ribosome, which is on the endoplasmic reticulum, and free ribosome in the cytoplasm is 80 Swedberg. So that's a 70S, 70 Swedberg. Right, then next one is the mitochondria. So mitochondria, so I'm going to show you the mitochondria structure. So mitochondria is again like this cigar shape, and they have two membranes. The inner membrane is folding into cristae like this, and that's a matrix. So outer membrane, inner membrane, that's called cristae, foldings, and inside again they have a ribosome and the circular DNA. That's a 70S ribosome and circular DNA. So if you look at the choices here given 1, 2, 3, 70S ribosome, 80S ribosome, circular DNA. So it's we have seen circular DNA, and then 70S ribosome. These two are common in both mitochondria and chloroplast. As a result of endosymbiotic relationship, that means the mitochondria and chloroplast, they were existing separately. Finally they went into the other big cells, and they used to have a symbiotic relationship, endosymbiotic relationship. So here we can see this one, question answer for that one, 1, 2, 3, 1, yes, 3 is there, 1 and 3, A is the answer for this one. So next question, Taesik disease, which is a genetic disease caused by some mutations, occurs when cells are unable to produce an enzyme, leading to a build-up of a certain lipid in cells. Which cell structure would not function correctly, resulting in a disease? So this disease cannot produce some enzyme. As a result of that, accumulate some lipid in the cells. Which cell structure would not function correctly, resulting in the disease? Okay, first one is Golgi. The basic function of Golgi is like this: Golgi cells, and Golgi going to get the protein coming from endoplasmic reticulum into that one, and basically protein modification is the function of Golgi, and after that by exocytosis they can either transport or they can use in the cells. And also Golgi produced lysosome. Lysosome. So function of lysosome, they contain the enzymes. Okay, enzymes. When I say enzyme, it's here. Produce enzymes. So enzymes cannot produce. They cannot digest the particles, especially the internal or external, like phagocytosis. The function of lysosome is fuse with the the pagosome, and then the pagolysosome, and they secrete enzyme like this directly into the particles, including bacteria, and they digest. So if there's Taesik disease cannot produce enzyme, lysosome has no enzymes, lysosome cannot produce function properly. So answer for this one is B. So we go to the next question, question number 5. Which structure are present in cell of Plasmodium? Alright, for this question, basically the chapter 10, the infectious diseases, we're going to learn the malaria. So we know malaria is caused by Plasmodium, could be vivax or maybe a falciparum. So what we learn from this chapter, Plasmodium, is eukaryotes. That's again one reason we can't make a vaccine, difficult to make vaccine as a complex structure. So they contain all the eukaryotic organelles, that means Golgi, ribosome and mitochondria. So it's quite clear, answer is A. We go to the next question, question number 6. The diagram shows a triglyceride molecule that has been partially hydrolyzed. Alright, so triglyceride molecule like this, we can draw OH, 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 H, 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 H. That's a glycerol molecule, and then the glycerol molecule, when they make a triglyceride, the COOH, so this one and this one, they're going to make a bond to make one water molecule, and the water molecule is removed. That's called condensation or polymerization reaction, and the bond is the ester bond. So likewise, they are making a ester bond with fatty acid, 3 fatty acid, just like this, 3 fatty acid, to make a triglycerides. And here already this one and this one is hydrolyzed. That means adding one water molecule and enzymatic reaction, we cleave this already this one and this one, and then this one is left here in the picture. If you see, this one is this fatty acid. So question is, what will be the product of the total hydrolysis of the molecule shown? Molecule is this one. If we hydrolyze, the ester bond is broken here. The ester bond is broken here. We get complete the one glycerol molecule and this fatty acid. So this fatty acid, you can notice here carbon-carbon, there's a double bond. So when the fatty acid has a double bond, we call unsaturated fatty acid. So this is unsaturated fatty acid, and then the glycerol molecules. So when we see this molecule and the question, better to save in time, we just go to find the correct answer. Here be a molecule of glycerol and an unsaturated fatty acid molecule only. So we don't want to read the other answers just to waste the time. So we directly pick the correct answer for this one. So we can save some time by doing that. That's a little bit of trick we had to practice when we're going to answer the 40 question within 1 hour. Okay, question number 7. Which shows alpha glucose? A very straightforward question. Again, as the previous question, we should not waste time to go through all the molecules. So I would really like if you can remember the structure of the glucose molecule. So it's like this. So carbon number 1, carbon number 2, carbon number 3, carbon number 4. Here is carbon number 5. So here you go. That's HOH here in carbon number 1 position, and HOH here. This is a bit different, OH here, and again same H and OH. So this one is only the black sheep, that means they behave differently. Otherwise they all HOH, HOH, H is up, HOH. So alpha glucose molecule, especially this position, H is up, OH is down. If it is beta molecule, OH is up, H is down. That's important to remember, and if you could able to draw this structure, I think we can save the time. So when you're going to see this question, just jump here. So directly you can see here, no, here, no, here, no, and here. The B1 you can see HOH, HOH, OHH, HOH, CH2OH. So answer is B. I think for this question, if you really know the structure, probably 5 seconds to 10 seconds you can answer this question. You can save a lot of time by doing that one. Okay, that you saved your time, and then this can be used to here because it's quite a lot of information in this question. Which correctly matches the functional and structural features of cellulose, collagen, hydrogen, or triglycerides. It's quite a lot. For my advice for this kind of a question, don't try to start from your left to right. So I suggest to go right to left from starting from here. So you can easily save some time, because most of the answers from the beginning you start maybe correct, and then finally the small incorrect answers are at the latter part. So we start here. So we have a cellulose, triglyceride, collagen, cellulose, collagen, glycogen, triglyceride. Okay, cellulose structural part, triglyceride is energy, collagen is a structural fibrous protein, both this collagen, and then cellulose is a carbohydrate, glycogen is storage, that's a lipid. So it's a mixture of carbohydrate, lipid, and the protein. So we start from this last one. Cellulose, branch chain. No, it's not a branch, it's make a linear structure like this. Cellulose microfibrils, which is joined by the hydrogen bonds to make the cellulose fiber. Triglyceride, we discussed triglyceride previously, glycerol and 3 fatty acid. So branch, nothing, no branch is correct. Collagen is again made by winding of a 3 polypeptide chain like this, triple helix, and then they don't have a branch here. It's not branch. Cellulose, branch chain. No, this is cellulose branch chain. They say yes. It's wrong, because this we can discard this, choice B. So same way choice C we can discard, because cellulose is a branch. So now we have A and D. So with the next one, molecule held together by hydrogen bonds. Cellulose, hydrogen bond. Yes, they have hydrogen bond. Triglyceride, they don't have hydrogen bond. Go to here. Glycogen, yes, glycogen is made up of 1,4 and 1,6. Like this, it's highly branched. 1,4 and 1,6. Like this. And starch and also glycogen, they make different kind of hydrogen, different level of hydrogen bonds. So they have a hydrogen bond, yes. And triglyceride, they don't have hydrogen bond. So this is also not correct. So the answer is this one, A, because we don't read these two, so we can save some time. So question number 8 we have done. So question number 9. Okay, the diagram shows the relationship between the levels of protein structure and bonds. So primary, secondary, tertiary, and quaternary structure is the protein, and they are bonds. So we just I want to show you a primary structure that has a peptide bond. Basically this is only, and the order of amino acids is the primary bond. Secondary, they make the beta pleated sheet and the alpha helix by making the beta pleated alpha helix, and also they have a hydrogen bond. And tertiary structure, they make a hydrogen bond, hydrophobic, ionic, and sulfide bridges or disulfide bonds. So they make hydrogen, hydrophobic, ionic, disulfide bond. So they make all 4 bonds here. They make a one type hydrogen bond. So they have peptide bond. And the quaternary structure is a combination of this tertiary structure like this with non-covalent bond to make the quaternary. It's a collection of several quaternary structure with non-covalent bond to make that's a quaternary structure. And they could also include all these 4 bonds because these are part of the molecule, and then maybe the ionic bonds or hydrophobic bonds are basically apart from the quaternary structure. The bonds between this is like these bonds, just showing red. These bonds are ionic and hydrogen or hydrophobic bonds they can make. Okay, so here now coming to the question, which row is correct? So we try to primary structure peptide. So again you start from the D. Quaternary structure, they can make ionic. That's a quaternary structure here. Yes, they can make ionic bond. And tertiary structure is this one. They can make ionic bond. Yes. And secondary structure, ionic bond. Secondary structure, they don't make ionic bond. So D is not correct. So then you can quickly try to find like this way: tertiary ionic, quaternary peptide, primary peptide, quaternary peptide. So quaternary peptide, this one, primary make peptide bond, yes, here. And quaternary, this one peptide bond not there, so we can remove this one. And the B, secondary hydrogen, this one, yes, the red one. That's a secondary, this one, red one. That's a secondary, they make hydrogen bond. Here, secondary hydrogen. Tertiary peptide bond. Tertiary peptide bond. Here, no, they don't make a here, peptide bond is not here. Primary peptide bond, that's correct, primary peptide. Next one, primary peptide, secondary ionic. No, secondary is not making ionic, and tertiary ionic, yes, they make it. So now when we read this question, they are very close to each other. So what we come to the now out of 4, D is closely related to the correct order, because quaternary, they make ionic, yes, and then tertiary,